Okay, so we're going to do our last note for this unit, and next week you're going to be doing some rather difficult research for me. So you're going to do a research paper. So we're going to finish up by looking at some of the genetic technologies that there are. First, you need a definition of what genetic engineering is. So that is the intentional alteration of a genome by substituting or introducing new genetic material. So literally, you're just taking the genes from one organism and putting them into the genome of another organism, which is something we've been doing for a while. An example of this is inserting the human insulin gene into the genome of a safflower. The safflower transcribes and translates the piece of human DNA to make the human protein insulin. So they started doing this with bacteria. So what they did was they took the DNA from a human that codes for insulin, which is a hormone, obviously a protein-based hormone since its code is in the DNA. And they inserted this into bacteria, and the bacteria were transcribing and translating to make human insulin. But now they actually plant crops of safflower because they've done this with a flower. Now, safflower does not grow well here, so we don't have any crops of safflower that are transcribing and translating human insulin, but where these plants grow well, they do have crops. And I'm not sure how they separate the human insulin from the rest of the plant, but I'm sure they have a method of doing it. So there's a perfect example of genetic engineering where you're getting a flower to make a protein that is from a human. And of course, for people who are diabetic, that is a great thing. Recombinant DNA is, if we look at the transformed safflower, it contains the two species genes. So if you look at its genome, it would now have human and its own DNA within its genome, and it's now called recombinant. So that's some pretty interesting stuff that they've been doing for a little while now in the field of genetics. And as long as you understand transcription and translation, you should understand that taking the piece of DNA from one organism and putting it into another isn't going to cause any problems with transcription and translation, since it just works like machinery. With the ribosomes just attaching, and they're just going to pump out whatever happens to be on that code. Restriction enzymes. So we have to be able to cut out DNA, pieces of DNA from organisms. And of course, we can't use scissors because the DNA is so small. But bacteria have these things called restriction enzymes. And these enzymes, their purpose was to chop up viral DNA to prevent them from being infected by viruses. But they also work great at chopping up any DNA we want them to. So we use them as molecular scissors. So they cut DNA at specific locations, and you end up with these pieces. The pieces resulting from the enzyme cutting the DNA molecule are called restriction fragments. So what scientists do is they use these enzymes to try and cut out whole genes out of a genome, because you don't want to be worried. If you just want to take one gene and put it into the genome of another organism, you need to be able to cut that gene out, and they use restriction enzymes to do that. So how restriction enzymes work, EcoRI is a very common one. And it locates, so it's going to cut in the same spot every time. So, and again, direction is everything with DNA. So it will cut 5' to 3' at GAATTC. So every time there is a GAATTC in a section of DNA, it will literally cut it. So, and this is how it cuts it. So you see GAATTC here. And again, at the bottom here, going in the opposite direction. GAATTC. So it will then cut, changes here, like so. And on the bottom, like so. So it will literally cut. And what it will have is these sticky ends. Okay, so it's cut the DNA. And now we can actually combine that with another piece of DNA with the same code here. And similarly here. So what you've done is you've literally cut that DNA molecule at a specific place. Do understand for gel electrophoresis, and this is the key component to how it works. Everybody's DNA order is slightly different. So everyone is going to have this specific code in that order different places in their DNA. And so when you apply molecular scissors to a person's DNA, it's going to cut their DNA in specific places that will be different from someone else's. So everybody's DNA will be cut into a different number and different sizes of pieces because not everybody has that code in the exact same spot. And that's how, then they use gel electrophoresis. So hopefully you did that little online simulation and you understand different size pieces will move differently in that gel. So again, I do not like teaching this without actually doing it in a lab. People understand it so well when we do the labs and we do the gel electrophoresis, but to do this on paper just does not do it justice. So I'm not going to spend a lot of time on it, but do understand that the different sized fragments of DNA are key in getting different patterns of DNA in gel electrophoresis, which happens to be over 99% accurate because nobody's going to have those specific codes that the enzymes cut in the exact same places. So there is your definition of gel electrophoresis. And again, we see these, excuse me, something caught in my throat. We see these different, bands showing different movement. So the larger the DNA, the less movement you will get. The smaller the piece, the more movement. And because everybody's pieces will be cut everyone's DNA will be cut in a different place, they will all have different size pieces. So they will all line up in gel electrophoresis at a different spot. The only persons that are going to line up exactly like yours are your own, unless you're an identical twin. So you might want to just get a definition of that. Again, you can look at these examples and see if you can make sense of them. And this is an important point you might want to take down. In paternity tests, 50% approximately of the gel lines will match with the true father and the other 50% with the mother. So that's how they do the testing for paternity with gel electrophoresis. So if we look at this example here is nice and clear, you got to figure out which male is the child's. So, understandably, half of them are going to line up with the mother. So this one lines up with the mother, this one lines up with the mother, this one lines up with the mother, these two. So we have these outliers that must line up with one of these males. So I'll let you figure out which male it lines up with, and that's how they know who's the daddy. Yeah. Crime scenes work the same way. You have your suspects, you do gel electrophoresis, the restriction enzymes will cut their DNA all in different places, giving different size pieces. And so when they phorese through that gel, they will end up in different places. So really clearly, there's the crime scene. There's the guy, right? Everyone else's are not lined up. So again, another practice question. These things called ladders are just there for measuring purposes. So when this is done in a lab for the purpose of paternity or for crimes, they have these things called ladders. So they can literally measure exactly how many base pairs the fragments are, and it's just more accurate that way. Another question you can take a look at. And that is it. So we're not going to spend any more time on the biotech simply because this is just a unit that should be actually practical, not pen and paper. It's just too hard to get your head around using pen and paper. So that will be it for your molecular genetics unit. We will be moving on to homeostasis next, but next week you are going to be doing some research.